our research work on the climate-friendly marine culture work. Today, the topic will be could climate-friendly marine culture be ecological trap? We have taken a case study from the mussel farming in the Mayan Islands. As we all know, the global climate change or the global warming has already affected the entire world. And IPCC suggests we have to achieve the carbon neutrality by the middle of the 21st century. Food production as a really important support for our human life, and also contribute significant to the climate change through the both directly and the indirect greenhouse gas emission. Because of the resilience on the natural resource, climate change posed tremendous challenges to continue the provision of the nutrients, security, and affordable food for the world's growing population. But not all the food is created equally in terms of climate impacts. The greenhouse gas emission per unit of the protein produced by agriculture are generally compared favorable with most livestock production. An updated estimate of the 245 megatons of carbon dioxide in 2017 were equivalent about 0.449% of the anthropogenic gas emissions, which account for more than 90% of global aquaculture production. Even though the aquaculture produced less gas emission, in recent decades, research further found that some aquaculture, like the algae or mussel farming, they could also capture the carbon dioxide and reduce the emission. Those aquaculture are therefore expected to achieve the food support and the carbon neutrality goals. Because both the algae and the mussel farming could bring the carbon bureau during the culturing stage, the seaweed and the bivalves was also called the climate-friendly seafood. In the 2018, over 7.3 megatons of farmed shelled mollusks, many will be marine bivalves, were produced worldwide, accounting for more than half of the global production of the marine and coastal aquaculture. According to the FAO database, global bivalves production has more than tripled in the last 30 years. So such a large volume and a rate of growth is therefore a pathway that could not be ignored in achieving the goal of carbon neutrality. So more and more studies will focus on the mussel farming. And some studies found that the mussel farms also have a really positive effect, like increased biodiversity. This is because the higher biomass and the complex three-dimensional structures that could provide the habitats and the food for the vertebrates and other animals. But however, there's also other studies suggest that the mussel farm also have negative effects, such as bring the noise, disease, and pollution, or other the different farming cycles will also repel some species from the farming areas, also cause the reproduction fails. Those negative effects will eventually change the community structure and the ecological function of the in those farming areas. And those negative effects will be considered as could be potential ecological traps for most marine mammals. As I mentioned in the previous studies, there's a different results about the mussel farming, and this caused really difficult to draw a conclusion that the mussel farming is good or bad for the biodiversity. So we in this study we used recently very popular technology, eDNA technology, to review the impacts of the mussel farming in the on the biodiversity. And here we hypothesize that if there's no difference in biodiversity, we think mussel farming was not ecological traps. Otherwise, would be potential traps for marine animals. To answer this, to test this hypothesis, we select the largest mussel farming areas in China, while it's the Ma-an Islands in the eastern part of China, belongs to Zhejiang province. Here is famous with the longline farmed mussels. And to test this, we conduct the experiment in the largest farm field in the islands. And the sample size was set in the 4 groups, which means in the central, the middle, the edge, and outside of the farming areas, to illuminate the effect by the DNA from the farming area. We also set another control set where it's far away from the farming area with the similar habitats. So in total will be 5 groups. Within the culture areas, we set the 1 kilometers away from each other from between groups, and also to reduce the more comprehensive results, we also set a triple replicates with 3 sample points within space between about 100 meters from each others. To collect the environmental DNA, we at each sample set, we collect 2 liters water samples from the surface, middle, and bottom layers. And after well mixed with the 3 layers, we only keep 2 liters water for the DNA extraction. Each 2 liter water sample were filled within 4 to 8 hours after collection by 0.45 microliter glass fiber membranes. And the membranes will keep at -20 degrees until the DNA extraction. The environmental DNA extraction will use the Qiaojin soil kits to isolate the DNAs. The concentration and the quantity of the extracted eDNA were examined using the Nanojob and the agarose gel electrophoresis. And to get a better answer for this, we choose 2 different types of animals to answer these questions. We select marine vertebrates and crustacean as a representative for high and lower mobile animals. For the vertebrates, we use the PR10 primer, which target 16 RNA and amplify about 202 base pairs. And for the crustacean, we use the MAD-DEC, which also target 16 RNA region for about 164 base pair. And after the PCR, we used the amplification. We used the 1% agarose gel to quantify the quantity of the PCR products, and the successful products were sent to the Personal Biotechnology company for the sequencing library construction and the Illumina MiSeq paired-end sequencing. The alpha diversity on the bioinformatic analyze were calculated to use the Chapman-2 for all the samples, and the long parametric tests were performed to verify the significance of difference between the groups. The MOTU, the abundance table, were analyzed use primary E software by the 2 similarity distance. The first one is the J-cut, which was unweighted distance used based on the MOTU present or absent. And the second one was the Bray-Curtis distance, which was abundance weighted based on the relevant abundance of each OTU. The two similarity distance metrics between groups were used for the beta diversity analysis. Many will be the long marriage multi-dimension scaling and the permutation multivariance analysis of variance. Significant difference MOTUs between culture and non-culture areas were identified by the DSeq2 package in R. After we do the analysis, and we found that when comparing the number of endemic MOTUs about different sampling groups, it was found that the percentage of the shared MOTU among different sample sizes was small for both vertebrates and the crustaceans. And the chi-square test showed that there's a significant difference in the numbers of endemic MOTUs among different sample groups. And in terms of distribution of endemic MOTU, vertebrates has shown the most endemic MOTU at the edges of the muscle cultured areas, and those endemic MOTUs are mainly from the perches. And although most endemic OTU from the for the crustaceans, on the other hand, we found that the outside of cultured areas has the most endemic MOTUs. Although most endemic MOTU was still really slow when considering the abundance, as we can show in the picture B and D. The abundance of shared was quite slow, especially in the vertebrates. The more than 90% were shared, but the crustacean showed a different tremendous, which was shared more than 90% in the cultured area, but in the non-farming area or cultured area, there's only 70% and 25% respectively. So this maybe imply that muscle farming has an inconstant effect on the different animals. When we look at the alpha diversity, we can show that whenever the number of observed species, chaotic Pineville, Shannon, and the Simpson index of nature are higher than the found in the vertebrates, but not in the crustacean. Even we have found those patterns, but when we do the non-parametric test, we found that the biodiversity indicators, none of them for both vertebrates and crustacean, they showed no difference, which means the muscle farming did not significantly impact the biodiversity in alpha diversity level. Also, to further explore the impacts on the community structure, we employed the PERMANOVA and the NMDS to find to review it. When we only considered the present or absent MOTUs, the vertebrates composition did not differ across the sampling set. We can see in the finger 3, the top left, they show they group together, and also the PERMANOVA, the checkout distance will show the p-value is larger than 0.5. However, when we consider the relative abundance, it is easy found on the finger 3, the top left. The control group with the green color is totally separated with the others, and our Bray-Curtis distance PERMANOVA will show the p-value is about 0.01, which was significant. And the similar results were found in the crustacean whenever it is based on the present or absent or the relative abundance. So this means that there is an effect on the muscle farming on the relative abundance of different species within the community. After we further look at the MOTUs between the farm area and the long farmed areas, and it is clear show that the different animals have different reactions. When we look at the vertebrates, in total we found 68 MOTUs in all the samples, and only one, the MOTU 10, I mean on the table, bottom of the table, it is showed a significant difference between the farming area and the long farmed area, and which was only found in the farmed areas. This MOTU is annotated as the Berlinidae, which was when we go through go through it. It is a small fish. However, among the crustacean, a total of 17 MOTUs in total is 87 MOTUs, was significant difference between the farm and the non-farmed areas. The 87 MOTUs, including 13 species. Unlike the case of the vertebrates, the MOTUs of the crab, I mean the crab MOTUs, which means from the decapods, was significantly lower in the farmed areas than the non-farmed areas. But MOTUs from the amphipods and isopods, which was a small crustaceans, were significantly higher than that in the farmed areas. Overall, the muscle farming areas have more likely to accumulate small animals. So based on those results, we can conclude that first, we think the EDN technology is really efficient tools to review the biodiversity. In the traditional way, it mainly relies on the visited fishing and diving or the underwater equipment to obtain the data, while the environmental DNA technology will mainly collect water samples and analyze the DNA composition of the water sample. So the environmental DNA technology has obvious advantage in the terms of the times, economic, and level cost in this way. And moreover, in these studies, because of the complex spatial structure of the facilities, it is difficult for the traditional survey like diving or fishing net, but it is not a big issue for the environmental DNA technology. Although the DNA has its advantage compared with the traditional, but it also have its own shortage. The main concern of the DNA technology is about the spatial solution of sampling, because a marine ecosystem is a dynamic system. The DNA will disperse from the sampling area to other areas. But in this study, and also the DNA will degrade during the dispersal, and the UV light, pH, and the water temperature also will accelerate this process. So we have the appropriate sample interval between some groups are the key to discover the biodiversity spatial differences. In the previous studies, it is common distance between the sample points were like up to 10 kilometers. In our study, we set the 1 kilometers between the groups and 100 meters between the sample points. So in the Primanova, we found that both for the vertebrates and the crustacean, between groups variance of the Primanova were larger than the residual variance, which indicate that the difference caused by the group were larger than the difference within group. This indicates that some distance in present study are able to review the spatial biodiversity. So in all, we don't think this is an issue for in these studies. And when we look at the impacts of biodiversity, Theoretically, when we introduced the aquaculture facility, which has provided the new habitats and also the food source for many wild populations, and therefore the aquaculture may increase the biodiversity. But in this study, we found different results. First, we as a famous mussel farming areas in China, mine has long mussel cultural history, more than half century. Also, it is reached the cultural area reached the 12 square kilometers. If there's some impact of mussel farm culture on the biodiversity, with considering the time and the area of culture in this area, it should be already have the some impacts. But as I reported before, there was no significant difference in biodiversity indicates of both the vertebrates and the crustacean between the farm and the non-farm areas. But in the vertebrates at the species level, they also find the same patterns. But in the crustacean, we found there are two groups are different from the have the received the influence from the first one is the large benthos in the Amaniwal crab in under the culture under the farming areas, and the small amphipods and isopods will increase the biodiversity in the farmed areas. This is because I think for the crabs, we think this long-term mussel farming has increased the sedimentation in benthelic the culture areas, which finally will cause the substrate types were changed and unfavorable for the crab to survive. Moreover, increased sedimentation will cause the increase of the ammonia concentration on the bottom, which will further reduce the benthelic fauna. And so we think the mussel farm has the inconstant effect on the different creatures, and it maybe have the relative small impacts on the swimming animals, but it will cause the great impacts on the benzos. Last, we want to answer the question: is the mussel farming an ecological trap? First, we found that the aquaculture theoretically will be, as mentioned before, will be have the attraction or exclusion effect on the wildlife. And our study will further support this. We found that some will be attracted and some will be excluded in the farming areas. But overall, we believe that the mussel farm do not have the significant attraction effect on the most animals and create a biodiversity hotspots. And this maybe imply that the mussel farm will be not an ecological trap for, at least for the vertebrates. And this maybe is due to, we think is because that the mussel farming areas is long feeding culture mode and do not provide directly food to the major of the vertebrates, and therefore they not attract the vertebrates and create the difference between the farming and the non-farming areas. For the crustaceans, we considering the dense and complex cultural stable the facility, I mean the structure may provide protection for the predation or the habitat which was relatively stable environment and a fixed structure substrate for the some crustacean, which may be attractive for the small animals, but for the relative large benthos lake, those floating rafts may not be attractive. So overall, there was no evidence in this study to suggest that a mussel farm is an ecological trap for either vertebrates or the crustaceans, except for maybe some small amphipods and isopods. But it is possible that the aquaculture may affect the abundance of some species of the crustacean, which may automatically eventually lead to the changes of the function diversity of the communities. But this needs further research to prove that. So let's summarize all the informations from this talk. First, we would think the environmental DNA technology is very efficient. And second, mass farm is not a great effect the alpha diversity, and the mass farming is not. We think it's not an ecological trap for most species, and the shellfish farming will be potential impact on the small and benthelic species. Okay, that's all for my talk today. Thank you very much.
Okay, thank you very much. Very interesting talk. But actually, in our faculty, we also have mussel farming in one station, so we should think about that as well. That's interesting. Anyway, so any question from the floor? Anyone want to ask the questions? Or even from online, you can turn on the microphone and then you can speak out. Or if you want to do that, you can type something on the chat box and I will read it for you. So any question for Cole? Okay, we have one question from our MC.
Yes, thank you very much for the presentation. Although I do not understand the details of the DNA analysis of yours, but I can interpret from the conclusion that mussel farming is friendly, right? It's not, it doesn't have any much negative impact to the environment. How about this one? Can you like extrapolate your result to the fish farming, for example, cage culture? What do you think? Will it be the same, or what? Can you say anything from your result?
Yep.
At the fish farming.
Okay, thank you for your questions. Actually, for the caged fishes, I don't do the research on this topic, but actually we have done a little bit of work on this, and we found that the caged fisheries were the case was significant difference from the mussel farming or the algae farming, because I think there's different types of the caged fisheries. Some will use the commercial depth for feeding, and some is used the small fish to directly feed the fish. And when we do the both, and we found this, the caged fish, the fisheries will attract small fish, especially the fingerlings, to provide food, directly food and the shadows for those fishes. But the same results will be the benthelic animals also be exclude for the farming areas, because the sedimentation, the farming is heavily increased the sedimentation in those areas, so the benthelic is heavily changed. I'm not sure did I answer the question for you?
That's perfectly. Thank you.
Right, Cole, can you hear me?
Yeah.
All right. Just a simple question extended from the Dr. Men question. Actually, the picture that you show us on the screen, this is the type of the mussel culture in China, right? It's like a hanging type, right? You use a net and you hang it in there, and when floating in the water, in the middle of the water, right? How about this in Thailand?
They use the long line. I mean, there's only a rope down.
Right, and you hang it, right?
Yeah.
And let it stay in the in a part of water. I mean, in the body of water, right? But in Thailand, we have a different system, another system. We use bamboo stick.
Okay.
We use bamboo stick and put it in like a fence or something when next to each other. What do you think? Will the result will be the same or different?
I'm not sure, but based on what I know, I think it will be similar, because although the facility, I mean the structure is different, but the animals inside, they have the same situation. They won't have directly food support, and they will provide the protection. Also because those facility and the mussel grows up here, and the sedimentation on the bottom will be also increased. So we should face the similar situation, I guess. but it will be have a chance to go through
Right.
the Thai culture model will be maybe have a better understanding.
Right. I think in the near future, you have to get you have to get your research team to be here, to study more about it, because this is the different type of culture. I mean, the technique, right?
Yeah, true.
One last question then. So
Yeah.
can we claim can we claim that our muscle culture is green culture, in your opinion? Can we claim it? Yet.
We have in China, we have a different opinion about this part because when the muscle grows, they think they produce the carbon dioxide into the atmosphere, so it is not pure green. But in my opinion, I believe it should belongs to kind of green. Green aquaculture, because it is non-feeding, and they also remove the nutrients and other organic matters from the water, and will at least will be environmental friendly culture model, I think.
All right.
Yeah.
Hopefully, in the near future, if you study more, maybe you can have more information, maybe a recommendation to set up a nice system and ready to be claimed as this is a green muscle culture to earn a positive credit or something. Right? Thank you.
Thank you.
Yes. Can I add a little bit one more thing? Another thing here. Mark actually talk about the muscle. There are a wide discussion group talk about how to reduce the carbon footprint in the shrimp culture, and one of the technique is to have a pond, like a wastewater pond, filled with the crab and mussel, and because the crab and mussel they accumulate calcium carbonate and also like carbon. So that's one like technique proposed by a fisherman and endorsed by the big company in Thailand, like Thai Union, that they would like to do that to reduce their carbon footprint.
Sorry, I missed the really beginning part of your question because the voice is not.
I'm sorry, it's not a question, but it is like adding to what you said about the green and not green as a muscle culture. I would like to say that in this morning there is a discussion about using clamp to reduce the carbon footprint in the shrimp culture in Thailand.
Okay.
So maybe it will show that at least in terms of the carbon neutrality, muscle is something good.
Yeah. Good to know. Thank you for your comments.
Thank you, Rashko, to spend your afternoon with us. And I know you are so busy at the moment. You need to move to another class. So thank you very much. Hopefully to see you in China, or maybe you come here to work on our mussel farmings, bamboo stick Thai culture. And thank you very much, Cole.
Thank you all. Thanks.
Okay. So next we will move to we're still in China. We will move to Professor Zhang Jihong. Professor Zhang is now the Deputy Director, the Yellow Sea Fishery Research Institute, Chinese Academy of Fishery Science. And today, Professor Zhang will talk about what I'm really interested in, because I'm also working on algae. So Professor Zhang will talk about the role of the macroalgae, that's a seaweed monoculture in the ocean negative carbon emissions. Please give a big hand to Professor Zhang. Professor Zhang, can you hear us at the moment? I think you need to turn on your microphone and turn on your camera as well. Okay. Professor Zhang, hello, can you hear me?
Can you hear me?
Yes, clearly.
Yeah.
Can you
Okay.
turn on your camera so we can see your beautiful faces?
Oh, it can't work. Just a moment.
Sure, no problem. Take your time.
I can't open it. Oh, sorry. Oh, yes, I can't find how to open the photo.
You can see the icon at the screen that they say to start the video. So I think you just click them and then
No. Oh, maybe you can hear me?
okay, that's all right.
Okay.
If you
Okay.
cannot, you can start talking and okay, we hear you clearly.
Yes,
Okay,
I
Professor.
start talk. Yeah. Hello, everybody. First, I will thanks for inviting me from the Kansas universities. Is right. And I'm Ji-Hong Zhang from Yellow Sea Fisheries Institute. My topic is the roles of microalgae mariculture in ocean negative carbon emissions. And my presentation have 3 parts. I will introduce the background quickly. Yes, as we all know, the global increase in the CO2 and other greenhouse gases that have driven the global climate change very seriously and caused a degradation of the ocean and the land-based ecosystem. So the global climate change is one of the most serious challenge to the sustainable development of human society. And as report from IPCC, the greenhouse that already released into the atmosphere is about 100 to 900 gigatons. So toward the worst outcome of the climate change to limit the increase of the global temperatures, the negative emissions technologies are needed to capture the CO2 that already released in the atmosphere. So the people pay more attention on the oceans. The ocean ecosystem is the largest capacity to sequester the carbons. And the negative carbon emissions based on the oceans are considered to be the important approach to control the temperature increase. And the nature-based solutions based on the blue carbon ecosystem to capture and store the CO2 and to reduce the CO2 emissions have received growing attention. The typical blue carbon ecosystem, with including the mangrove forest, salt marshes, and seagrass based. And although the natural seabed forest has not been included in the blue carbon ecosystem, but there are a lot of reported on its carbon sink functions, such as is the barrel rate of the seabed organic carbons in the sediment. It accounted for nearly 10% of the seabed productions. And this is study on the green tile in China, in Qingdao. They have studies of the long-term degradation process. About 1.6 of the green tile seaweed is biomass carbons will live in the seawater in the form of the RDOC. R is the reflector dissolved organic carbons. RDOC is highly stable and can be stored and accumulated in the seawater for million. It's a long time. We can consider it as a carbon sink. But under the influence of the multi-pressures such as the environmental pollution and the coast energy and the climate change and others, the natural seaweed forest declines is serious. So people have pay more attention on the seaweed mariculture. This is the current status of the seaweed farm in the world, and it's from the FAO, the year of 2020. The seaweed that represents over half of the total production of marine cultures, and over the 80% of seaweed farm are focused in Asia. For example, in China, in Indonesia, Philippines, South Korea, and so on. China is the largest mariculture country in the world with a seaweed yield of about 2.7 million tons. That is the sunlight dry weight. So the seaweed farming have a lot of benefits, such as it don't occupy the land resource and needing the freshwater resource. Also, no need to feed, to fertilize. Also, the seaweed can grow fast, have the high carbon sequestration efficiency. Also, the culture industry of the seaweed is developing very quickly. Also, they have a lot of potential species that could be captured. It's over 12,000 species. Also, they have a lot of sea areas that are suitable for farming. So, seaweed farming has a very potential benefits. As a primary producer of seaweed, its carbon sink function has attracted worldwide attention, especially in recent years. They have some published papers on this. This is potential carbon sink pathway of farming seaweed. Seaweed can encourage the oceans to absorb the CO2 from the atmosphere by the biological pump and the solubility pump. Part of the gross seaweed will be harvested by the production. Also, the seaweed can lose and release some POC, particle organic carbons, that will be buried in the local sediment or exchange to the ocean, to the open oceans. Another pathway is the seaweed will release dissolved DOC, and by the microbial pump, they will transport the DOC to the RDOC. I will introduce this potential pathway of farming seaweed. This is the second part of my presentation. First, I will introduce the biomass carbon in the seaweed. In 2005, we published our papers, and that papers we pay all attentions on the contribution of the mariculture, shellfish and seaweed on the carbon cycle in coast ecosystem. We calculate the biomass carbon in the seaweed according to the annual yield of the seaweed and the carbon content in the seaweed. Also, we published papers in 2011. We calculate the 10 years total carbons that be removed from the coast ecosystem by harvest. And then from then on, China have provide a new conceptions. We call it a fisheries carbon sink. And recently we study on the buried POC and RDOC from the farming keep. We studied the case study in Sango Bay. I think Sango Bay is a typical suspended long-life culture areas in the northern of China. Near the coast, we catch the bivalves, and in the middle of the cave of the bay is a polyculture of the bivalve and the seaweed, and in the outer side of the bay it captures the cave. So we use the ocean pike flow through system. Also, we collect the surface seawater to measure the pressure of CO2, and we collect the surface sediment to and the sediment column samples. Also, we do some in situ experiment to measure the POC released and the RDOC production. And some I'll not introduce the detail on the method. And first, I will introduce the CL interface carbon flux. This is the formula we calculate, how to calculate. And some result from the 4 seasons, from the spring, summer, autumn, and winters, and from different culture areas, bivalve areas, polyculture areas, seaweed area, and control areas. And from our result, we calculate the pressure of CO2 and calculate how much of the CO2 exchange between the air and the sea interface. This is some result. We can see the seaweed farming areas become the stronger carbon sink. Is the interface carbon flux is some result. I think for the different environment and the different seaweed, the result is different. So I not tell the detail on the date. And this is the annual absorption of CO2 of the total bay. And the second, I will introduce another pathway of the carbon sink of the farming seaweed. That is the POC derived from the cape. The cape is captured in the long line, and the POC will lose from the cape during its growth. There are 3 factors for the loss of the seaweed. One is the remove of the seaweed from the culture rows. Another is the breakage of the seaweed. And the third is the erosion of the distal tissues. So we calculate how much of the seaweed will fall off from the rope, how much will break off during its growth cycles. Also, we calculate how much of the POC from the erosion. We calculate the erosion rate of the cape. Is the red line is the production, and the blue line is the erosion. So at the summer, during the summers, they will erosion very quickly. So this is the result on the total loss of the total production of the POC from the seaweed. and this will occupy about 45% compared to the gross production. This result is very high. Another POC release during the seaweed growth is the seaweed will release very small particles during the seaweed growth. We measure this in the experiment by the also general experiment. We measure we use the different levels of temperatures, light, and the photoperiods. We got some result, the release rate of DOC and the POC. Comparing the release rate of POC that is in this range is lower than the DOC, but also as the seaweed grows, we could release the POC into the seawater. It's very important, I think. The third, I will brief introduce the carbon derived from the debris that are buried in the sediment. We calculate the surface sediment, and we measure the stable isotope of the carbons and the nitrogens. We using the mixed zero models to calculate the organic contribution from the phytoplankton, from bivalve, from the seaweed, also from the terrestrial organic carbons. And this is some result that published in this year. We can find about 30% of the surface sediment organic matters that come from the Cape. I think the seaweed culture was the major component of the sediment organic carbons in Sango Bay. Also, we measure the pump radioisotope activities to calculate the sediment rate and the burial rate of the POC in the sediment. This is formulas, and this some result. I got the deep sediment core, and then we calculate the sediment rate from the shellfish arrow, co-culture arrow, and the seaweed culture arrows, and this some result from. Also, according to the organic source in the corn, we could calculate the organic matter was mainly come from the marine source. Also, we can get how much from the seaweed, from the biodeposit of the bivalve. And this is the burial rate of total organic carbons and the total nitrogens in Sango Bay over the about 200 years. From our result, we can see as of the long-term culture of the seaweed, a lot of organic carbon will buried in the sediment of Sango Bay. Comparing with other bay, also in the north of China, the Sango Bay, it's a typical culture areas. The seaweed mariculture will accelerate the water and the sediment interface carbon flux and contribute to carbon borrows. This is some result of total organic carbon. Under the fourth, I will introduce the RDOC. DOC released by the sewage that is a critical component of the coast oceans biogeochemical carbon cycles. Recently, there are two passes, on the DOC release. There is the passive release, such as the leakage that requires no energy. Another is the active release that will require energy, such as these figures. And the release, the DOC, will form the RDOC by the microbial carbon pond. We did the experiment in situ in Sango Bay after long-term experiment. It's about over 440 years. The DOC was converted to RDOC over 200 days. Also, we calculate how much of the RDOC will produce. If this data is unpublished data, the paper is under review. And this also in Sango Bay, and we calculate how much of the ratio of RDOA to the net primary production, also the ratio of RDOC to DOC. This is some mini result. So the third part, I will go to the summaries and prospects. From our result, we think the mainly contribution to the carbon from the sea farming that includes the seaweed biomass carbons. That is the seaweed, the carbon storage in the seaweed that removed by the harvest. Another is organic carbons in the sediment. And the third is the RDOC. Also, for the seaweed mariculture is very complicated process in the ecosystem. It's including the interaction of organic and inorganic. It's interaction biotic and abiotics. Also, it's a lot of exchange between the water and the sediment, atmosphere and water, and the marine ecosystem and the open ocean ecosystem. And most we should, when we consider the carbon sink of the farming seaweed, we should apply the negative environment impact, and we should keep the seaweed farming in the sustainable development. It's very important to base on the carrying capacities. What we'll do in the future, I think, I have research on this natural carbon sequence pathway. In this mariculture stage, we all know for the seaweed farming is life cycles from the seeding stage to mariculture stage to the harvest, storage, transport, processing. Is as these humans activities, they will spend the energy, some materials that will release CO2 into the atmosphere. So when we calculate the carbon sink, we should also pay attention on the carbon footprint. The second, we should pay attention on the seaweed that we harvest. If we use the seaweed as the animal feed, the fertilizers or the bioenergies, that will is a environment friendly ways. It's a carbon alternative pathway. It's very important, and there are some reported on the harvest waste. The waste of farm seaweed that is used determines its role in the carbon cycles. Also, if it as the food, the biomass carbon in the seaweed that were released in the atmosphere in the quickly, we call it a fast carbon cycles. If we use the harvest seaweed in the form of the fertilizers or bioplastics or other ways, that is a long-term sequence for the carbons. So we call it the slow carbon cycles. So it is very important to care more attention on the carbon footprint. Some report, if it's like the animal feed additive, the carbon footprint, you have a great change from the negative to the active. That means is maybe is a carbon source or the carbon sink is very important. So the third work that we need to do in the future is to set up the technical of practice for how to investigations and how to assessment on the carbon sink of the mariculture seaweed. They until now they have international practice, international technical code of practice for the mangrove forest, salt marsh, and the seagrass species from the CDMA or from the VCIs, but still they didn't have the international standards for the seaweed, especially for the seaweed farming. So this is a WANS international program that has set up to cooperation research on the ocean-based solutions for the climate mitigations. The WANS international program the mainly work there, including the seaweed farming. That's a very important part of the WANS. So this is the website of the WANS, and there are a lot of countries and colleges and research unit join the WANS international program. If anybody are interested, we are welcome to join this the WANS international program. Also, in the for the WANS program, they have set up a work group 15 in the ISO. It's a TC8. The meanings the aim of this work group was to set up the international standards for the not only the seaweed, for the oceans negative carbon emissions, to measure the carbon sink measurable, reportable, and verifiable. That's very important for the trading, for the market, for the seaweed farming to get more monies from the market, for the carbon market. So this is my name card. and hope cooperations with US. And okay, thank you for chairman. Yeah.
Thank you very much, Professor Chang.
Yeah.
So it's really interesting topic, and this kind of the presentation that kind of reconfirm my interest in seaweed. That seaweed is kind of very wonderful marine species that can be very benefit in all kind of things. So any question about the Professor Chang will be with us and ready. Happy to answer. Oh, we saw you now. Finally. Okay, so any question from the floor or from the online platforms?
Hi, this is me again. Wow, it's I would say it's a very eye-opening, because this morning we discussed a lot about how to achieve the very ambitious carbon neutrality, because in the world in year 2050, right, we have to achieve that. And I can see that there will be two ways to do that. First is to reduce the emission, which is almost impossible, and the second is to capture the carbon. And to capture the carbon, they talk about many things, and they talk about the carbon credit from the forest on land, and also for the blue carbon. But according to your presentation, and I also asked the same question this morning, that seaweed and also actually the plankton, the phytoplankton, it can be source of the big carbon sink. So can I have your idea why we cannot add them as a blue carbon? Why? What is the difficult part? Why we cannot make the seaweed as part of the blue carbon? Thank you.
It's a good questions. First, it's very difficult to qualify the carbons for the long term. One is for the harvest seaweed, if it as the food will rebuild the carbon, the CO2 in atmosphere. So it's a lot of questions. For the RDOC, still they didn't have a standard method how to measure and calculate the RDOC. In my experiments, we measure these experiments in over 240 days. But I think it's not a good way. Still, they didn't have a qualified method. And another way for the POC in the segment, it's still very difficult to qualify in the open areas. How much will transport from the marine culture ecosystem to the open oceans? So that still we need to set up the international standards. I think if we get it, it will be included in the blue carbon ecosystem, I think.
All right, thank you. Just to continue the question, yes. I think your result, and also you mentioned about many reference, that it is clear scientific evidence that it can be a good carbon sink, but the methodology is still establishing, right? So what do you think? It be soon to be able to include the seaweed, or it cannot be forever? What do you think? We expect another 5 years, for example, for the seaweed to be part of the blue carbon.
The signal is not very well. Can you pardon speaking? Can you pardon your question?
Oh yes, I ask again. I know that there is a scientific proof that the seaweed can be a good carbon sink.
Yes.
The difficulty that it cannot be part of the blue carbon because of the methodology, right? Difficult to measure. But if we continue improving that, how long? What do you think? How long will it take for the seaweed to be part of the blue carbon internationally?
Yeah, yes, I'm not sure, but from the WISE international program it is one of the work we'll do to set up the international standards. I think it maybe at least 5 years. I'm not sure. In China, we have set up the international standards, not the national standards. I think I have to move step by step.
Yes, but it's coming, right? It's coming.
Yes.
In our lifetime, we will see that seaweed will be part of the blue carbon. Thank you.
Thanks.
All right. Dr. Chang. Dr. Chang, you hear me, right? You can hear me, right? Okay.
Okay.
I just extend my comment from Dr. Napong. China is a big country, right?
Yes.
It's almost the biggest one, as far as I know, right? You have a measure in like economy leader or something. Right now, for the part of seaweed, you are the major producer and exporter for seaweed, right? Number one for the world, right? The second one, I think if I saw your graph, I mean, correctly, the second one will be Indonesia and the third will be Philippines, right? China is number one.
Yes.
Right? Major producer for seaweed. I think right now we are talking about international standard. Mainly it cleared by maybe some other country in Europe, maybe in United States somewhere, right? Why don't China? Because your country is big enough to call it out loudly. I mean, you can set up your own standard. You can be the leader in terms of this kind of thing, right? Don't just wait for them. Just wait to standardize with them. Why don't you just create your own standard? Chinese standard. In order to have to clear the problem about carbon credit, blue carbon credit or something. I think you have a lot of opportunity because you are the big country, very, very big country. So move forward. I mean, move on. We are right behind you then, right? Go for it. Go for it. That's my comment. Thank you.
Thank you very much. You want to say something, Professor Chang? Are you thinking about you are now thinking about set up your own Chinese standards for this kind of thing? Okay, because we know that China is a big country. I mean, the major producer of Hai Tai. I think that's we are really interesting to see if China can step a little bit forward to establish something like this. And actually, I just heard from like a few months ago that we had a meeting in some country in Europe about I think UN organized a meeting and talking about the blue carbon, and there are some people speak about the seaweeds there as well. But anyway, yeah, we just need to wait. But anyway, as my co-moderator said, we will support China anyway. Okay, thank you very much. I think time is up. So thank you very much, Professor Chang. And if you still have time, you can stay with us until the end of the conference, because I think they might have some question pop up along the way. So thank you very much.
Okay, thank you.
Okay. So the next ones, the first we're talking about the aquaculture. So the next one we will go to the something ecosystem ecology. We have Dr. Milika with us. So actually, frankly as well, I read some of your publication about the blue carbon and sea glass, and we know very well that you're working on this kind of things in Trang Province or in River Island, I think. So now very happy to have you with us today to give a talk, and I think this is a hot issue at the moment about the blue carbon. If you are ready, we will pass this stage to you. Please give a big hand to Dr. Milika.
Thank you. Thank you very much for the invitation, and my apologies that I cannot be there in person. Before I start the presentation, you mentioned the UN meeting I was invited for that meeting. So there is a talk for the seaweed and incorporation for the blue carbon. But the biggest gap that they found is the methodology, as Professor Zhang said, and the data is missing for many regions. So there will be a publication coming up probably this year about that, about the future of the blue carbon, so the updated. So there is a specific question related to the seaweed carbon, just to give the heads up. Okay, I will share my presentation right now. You can see the full screen, right?
Yes.
Okay, great. So go. Okay, so from the macroalgal blue carbon, I'm just going to move forward to the traditional blue carbon systems, to the mangrove forest and seagrass meadows. I'm not mentioning the salt marshes or tidal marshes because we don't have them in this region. So my work is mostly based on the seagrass meadows and a little bit more for the mangrove forests. So as we know, both mangrove forest, the ecosystem services of mangrove forests are multiple, and they benefit not only for the climate change, but also for the livelihoods of the species, the tourism, the coastal protection, as well as the food that we eat. The similar thing is for the seagrass meadows, where we have dugongs and turtles feeding off the seagrass. We also have the climate change part of the carbon storage and sequestration, but as well nurseries for many fish and other invertebrates. So the globally looking, we are talking about 70 seagrass species, which cover about 159 countries on 6 different continents, and roughly they cover about 0.1% of the ocean floor. And we can see that Southeast Asia, so where we are, is the hotspot in terms of the biodiversity of seagrass species. When we look at the mangroves, also globally, they are more located in tropical and subtropical region in more than 100 countries. And again, Southeast Asia is a hotspot with more than 50 species of mangroves occurring in this region. And both of the speakers before me mentioned about the climate change and the need to reduce the emissions. So we have the natural sinks in the nature that we can use to mitigate the climate change effects. So the gray and the orange, let me just okay. So the gray arrow and the orange arrows are the fossil fuel emissions, which are quite high. From the gray one is the anthropogenic fossil fuel emissions, and the orange arrow is from the land use changes. And we have the natural carbon sinks, such the green arrow, which is the terrestrial carbon sink, and the blue arrow is the ocean carbon sink, and we can see that these are very large numbers, especially the ones that we have in the ocean. So when we compare the burial rate of the carbon between different types of ecosystems, compared to the coastal or marine ecosystems and the terrestrial ecosystems, we can see that there is a high difference in the carbon burial rate between these different types of ecosystems, where the coastal ecosystems such as salt marshes, mangroves, and seagrasses store much higher much higher carbon accumulation rates than the terrestrial forests. And basically the carbon within that is called the blue carbon. So the carbon within these ecosystems is mostly stored in the soil, which is, we can see the brown color, compared to the terrestrial forest, where the carbon in the soil is much lower. And in the terrestrial forest, the carbon in above ground biomass, in the living biomass, which is green color, is much higher comparing to the blue carbon ecosystems. So when we have a terrestrial forest with the majority of the carbon in the above ground biomass, when there is a fire or there is a lodging or land clearing, a lot of that carbon gets released back into the atmosphere. But when we have mangrove ecosystems or seagrass ecosystems, only a small portion of that, only small portion of the carbon that is within the living biomass gets released back into the atmosphere, while the majority of the carbon that is within the sediment is slowly being released back compared what we have to the terrestrial forest. So I'm saying that basically when we cut down the mangrove forest or dredge the seagrasses, the carbon that is being trapped in the sediment is still gets released back into the atmosphere, ocean, and then the atmosphere, but at much slower rate. So when we look about in Southeast Asia, the seagrass area is about 6 to 12% of the global seagrass area, with the highest areas in Indonesia and the Philippines. And the mangrove area is about 32% of the global mangrove area, where Indonesia is leading with Indonesia with the highest area of mangrove forest within all the region. When we looked in terms of the blue carbon science, so when we wanted to see the overview, who is doing what and what is missing in the region, we can see that a lot of countries are working on the mangrove forests, such as India and Indonesia, with quite a lot of published studies. And for the seagrass meadows, we have only Indonesia with 31 studies and Thailand with 29 studies. The rest of the countries are really not working on the blue carbon in the seagrasses. So yeah, seagrass data is missing from majority of the country, which is very concerning, especially as this is the hot spot in terms of the biodiversity and very high data deficient region. And then we have the countries where there were no studies on seagrass at all, such as Bangladesh, Sri Lanka, or Timor-Leste. I think Sri Lanka and Timor-Leste are working on it now. Bangladesh, not yet. And we found the areas called blue carbon hotspots, where the carbon was higher than average compared to all the other locations, all the other reported locations. So from 140 locations across all region, we identified the areas where the carbon was above highest. And for the mangrove in Thailand, we identified Trat River, the mangrove area around Trat River, as the blue carbon hotspot, and Libong Island when it was healthy. Just have to note that as the blue carbon hotspot. Across the region, for the seagrass carbon stock, it's about 35% of all. If we look at the total blue carbon in Southeast Asia, seagrasses store about 35%, and compared to the other regions, the seagrass in this region is about 1 to 5% of the total global carbon stock. So it's not the highest, like compared to Australia, but it's still much higher compared to the Mexico, where it is also the same region, similar region, and similar constitutional species. When we looked at the Southeast Asian total blue carbon stock of the mangroves, it was much higher in all the countries across the region, in Thailand as well. and it was about 65% of the total blue carbon. So a lot more blue carbon is being stored in the mangrove forests compared to the seagrass meadows. We also identified how much is the stock. We estimated the stock for each country. So for example, the highest mangrove stock was in Indonesia, followed by India and the Philippines. In Thailand, it's also it's mediocre, I would say. It's above 1,000 megagram per hectare. But when we look for the seagrass meadows, we have India as the country with the highest total carbon stock per hectare, followed by Thailand. We also looked at different national blue carbon stocks. So, for example, if we want to use for example, when you're setting the carbon markets or international frameworks, you have to know how much you have within your country. So we assess the literature that has been already published, and we estimated that, for example, in a living biomass above ground for both mangroves and seagrass meadows, this was the average stock. For below ground carbon, it was much higher for both mangroves and seagrasses. And for the sediment, it was up to 500 megagram of carbon per hectare for mangroves and up to 200 megagram of carbon per hectare for seagrass. Of course, there was some data available for the mangrove forests deeper than 1 meter. And if we go deeper than 1 meter, the carbon stock increases much higher. And I think the deepest core that we looked at was about 3.5 meters. And the carbon stock was almost 1,500, 600 megagram per hectare. But usually for all international framework and policies, 1 meter is considered as a standard. Furthermore, we looked at how much carbon is being buried within the ecosystem. So for the mangrove forests, it's more than 1,000 grams of carbon per square meter per year, while for the seagrass meadows, it was about 3 grams. But I have to know that this is a single study, and we are missing a lot of data for this. So I wouldn't say this was the national average, just what was available. And we are working on this to incorporate more studies so we can get a better estimate for the national carbon burial rate for the seagrass. However, we know that these ecosystems are being lost. For the mangrove ecosystems, a lot, so the region F, majority what has been lost for the commodities, which is conversion of mangrove forests to aquaculture, agriculture, and similar, and erosion is another part. However, when they analyze the global mangrove loss up to 2020, the researchers concluded that the net change has decreased significantly, and so far within these 20 years, it is about 0.16% per year. So we are not losing we are losing less mangroves than 20 years ago. However, there are still hotspots where the mangroves are being lost, such as Caribbean. This is Myanmar, and this is Indonesia and India. Thailand is not there. However, we are losing the seagrass. So this is the picture from Libong Island from 2016, 2021, and last December 2023. 2016 was actually start of my PhD. It was all green and very nice looking seagrass meadow. And then when we started revisiting it for other projects, we started taking photos of how the meadow has been changing. So we have a series of collage images of the meadow changes across the year. And we can see there is some dramatic changes even with these several years. When looking on the global scale, from the 80s until 2016, there has been a net change of seagrass of about 19%. So quite a lot of seagrasses have been lost. However, there are data deficient region, which is our region is one of them. But the recent study from 2021 estimated that about 4.7% of seagrass have been lost between 2000 and 2020, which most likely is higher now because we are experiencing quite a large seagrass loss in Thailand. So we are reassessing that. And this study concluded that the threats are coastal development, sedimentation, destructive fishing, and aquaculture. And these threats, the source of the threats has been the same as 30 years ago. So it is kind of sad to see that nothing really changed in terms of the management of the coastal area. So what happens to the carbon? So we have a healthy seagrass or healthy mangrove, which is a carbon sink when we have degradation or complete loss of the ecosystem. That carbon starts to be released back into the ocean and then consequently to the atmosphere. In one of my recent studies, we kind of looked at the changes at Libong Island. So this is until 2019, so before dramatic changes. The red area indicates the loss. Indicates the loss of the seagrass. The blue area is the stable area of seagrass, and green is the new area, so where seagrass expanded. And across these years, we estimated that there was a loss about 3.2%, which right now, from 2019 until 2024, is most likely higher. And we are working on it right now. So these are the series of the seagrass drone images from one of the area at Libong, where you can see at 2018, you had a healthy meadow and Hallos acroides with different mixed seagrasses. This was dense and Hallos and dense mixed seagrass. And by 2023, which was November last year, in some areas, there was just only Halophila left. There was dead rhizomes of Enhalos, or there were some areas with no seagrass. So it's not only that Libong is experiencing this, but other meadows around it. So we are trying to quantify these changes across different types of meadows. So for example, at Haja Mai National Park, the study from 2018 found that the seagrass meadow there has been stable since 1973. So the seagrass hasn't changed in over 20 years. And then when we went on the first project to collect some data in 2021, it looked very nice and healthy. But then in 2023, the areas where were very densely populated seagrass, there was nothing left, or there is only Halophila or with dead and Halos roots, or it was very sparsely distributed Halophila. So the seagrass meadows are definitely changing a lot across many regions of Thailand coast. I also heard that a similar thing is happening in the other countries in the region, but haven't been quantified yet. So we're definitely not alone. but there is definitely something going on. So as I mentioned, when we lose the ecosystem, the carbon that is being trapped gets released back. So we roughly calculated, so these numbers are before the major changes that we are experiencing right now. So we roughly calculated that the potential emissions were between 0.5 and 2 teragrams for each year from the seagrass changes, which is basically equal to 500,000 of cars, emissions from 500,000 cars. And when you also lose the seagrass ecosystem, you not only lose you don't only lose the carbon that is being buried there, but you are also lacking the sequestration. So the potential of how much carbon would be buried there, trapped and buried there. So we calculated that is additional almost 700 gigagrams of potential sequestered CO2 for every year. And that all accounts for over 20 million, loss of 20 million US dollars in terms of the market value. However, we are still, so this is from the study from 2021, but we are currently updating this and working on how much is working on the studies to estimate really how much of the carbon is being released from degraded meadows or completely lost meadows. So I think in this year or next year, we'll have much more, we'll have more better estimates of the losses and the potential of the seagrass. So I will just go back a little bit and explain that for the Paris Climate Agreement, every country has signed nationally determined contributions, or NDCs, which are basically their pledges of what they are planning to do to reduce the emissions, on the country level. Based on that, each country has developed their business as usual emissions. What would have been in, for example, 2030 if no pledges for climate change mitigations occurred. So this is example for Indonesia, Malaysia, Thailand, Vietnam, and Philippines. And again, these numbers vary from country to country. And from last year, no, the year before, we have a new set that everyone is trying to reach, a carbon net zero policy by 2015, where all the CO2 emissions are going to be offsetted by some kind of carbon capture system, which is going to be natural or engineering. So within that scope, we are looking how much of seagrass and mangroves can help with that too. So currently, there are a lot of countries that are including the blue carbon ecosystems, mostly mangrove, but there are some countries that are including seagrass as well into their national policies. So all the blue countries like China, India, Indonesia, Malaysia, Vietnam, USA, they have included some kind of form of blue carbon into their national agreement. The good examples that I would like just to highlight are Belize, Sri Lanka, and Seychelles, which not only included mangroves but also seagrass ecosystems. And they have included blue carbon into their greenhouse gas inventories and are planning to conserve and restore these ecosystems to help mitigate their the climate change effects and offsets for their countries. And then right now there is a next round until next COP where the NDCs are being revised. and many more countries are looking into including both mangroves and seagrasses into their greenhouse gas inventories. So when we look at mitigation potential, so for both mangrove and seagrass, there are two options. So we are looking at the business as usual, where the ecosystem would continue to decline, even with the natural fluctuations. And the other option is what we are looking at is the additional carbon stock. Additional stock with the increase of the ecosystem in different areas, the same area where it existed, and so on. So there are two different type of projects, I would say, that are possible to do through this. The first one is the conservation, and the second one is the restoration of the ecosystem. So in the conservation, we already have established ecosystem where, however, it is being degraded as the time passes, and the project would come and increase across the years. It would basically stop the decline and improve the ecosystem. So in terms of conservation, we are accounting for the carbon through avoided emission. So what could have been lost, and the loss of sequestration. In the restoration project, we are looking at the ecosystems that have been completely lost, and they need to be restored from the beginning. And in there is the new accumulation of the carbon is being looked at. In most of the cases, restoration takes years, especially when we are talking about seagrasses, because a lot of the carbon is being stored in the sediment, while the conservation, you have options for both. If we look globally, the potential for avoided emission, so conservation approach, it varies from country to country, but you can see that the region of Southeast Asia has a very high potential for avoided emissions through mitigation projects. And if we look in terms of the types of ecosystems, mangroves, tidal marsh, and seagrass meadows, the avoided emission is the blue color, while the restoration can add on 10 times higher rates of carbon capture within the ecosystem. However, we must look at it a bit conservatively, because the restoration takes years, while when we do the conservation and avoided emission, the impacts on the ecosystems are in much shorter timeline. So for the restoration potential, there are some current global declarations. For I think for the tropical forest is about 3%, for peatlands is about 5%, and from the last COP there is mangrove and seagrass breakthrough, which are planning to restore about 20% of the mangroves and about 30% of the seagrass meadows. And if you account the carbon burial rates of these new seagrass and mangrove area across the countries, by 2030, for example, in Thailand can rely very heavily on the mangrove carbon sediment capture and the seagrass, while for terrestrial forests and peatlands, it's much lower. So these are the values for the sediment. The additional one would be for the living biomass. So what does it mean in terms of overall looking for the country and net zero plans? So we can look these pink squares, which represent the NDC range targets. So it is how much of CO2 emissions every country needs to decrease. And we briefly calculated that seagrass and mangroves, depending on from country to country, can contribute 6 to 7% for these NDC targets. So far, from our previous studies, we calculated that both seagrass and mangroves can contribute between 0.5 and 1.6% of the NDC target. It sounds very small, but the estimates and the values that we used for the calculations are, I would just like to know that they are not final. For example, we don't know the rate of the seagrass decline yet, the most current one or the most accurate emissions across the region. So I would say these are the conservative measures, but we are working on updating this. However, both mangroves and seagrass meadows are most poorly represented ecosystems for climate change mitigation potential. And across globe, there are very few projects that are looking into offset, using the carbon for offset from both mangroves and seagrasses. So seagrass would be the blue color and the mangrove would be the green. We can see for the seagrass, we have one project in USA, and we have several projects in Japan, and then in Kenya, I think, for the seagrass as well. So a lot of countries are heavily relying more on the mangroves, while the seagrasses are being used only in several countries, although the methodology, the global standard and methodology, like Professor Zhang mentioned, exists for both of the countries, but the data and the national awareness of the seagrass is much lower compared to the mangrove forest in many of the countries in the region. And of course, I would just like to mention that we shouldn't forget the other ecosystem services that all both blue carbon ecosystems provide. They improve water quality, they provide coastal protection, which is climate change, which adds on for the climate change adaptation strategies. They improve fisheries and secure food security, and of course, biodiversity. And all these benefits from all the other ecosystem services not only contribute towards the Paris Climate Agreement goals, but for the various SDGs, Ramsar conventions, and also Aichi biodiversity targets, which when we are looking, when we are incorporating these ecosystems into international policies and potential what they can do, we can pick off many different national goals for each country. And that would be it for me. So thank you very much.
Okay, thank you very much, Dominica. Very interesting. We see some number. That's very interesting number as well from your research. Any question from the four people in online platform? You can open your microphone and you can ask, or even you can type your question in the chat, so I will read it out. Anyone want to ask any questions about blue carbon? Okay.
Yes, it's me again, Tanas Pong. I'm very interested.
Thank you.
Yes, I have a very generic question because you present a lot of information about how Thailand, how the regional, and how the world study about the blue carbon, and also also the progress of the blue carbon research. Okay, I have a simple question about the blue carbon things in Thailand. Maybe you also involved in some of the giving the information or seeing that in Thailand seagrass plantation. It's really a hot topic now, especially for the big company like oil company. They always go for the seagrass plantation. Can you say something about this? What do you think about seagrass plantation? Is there any other ways to improve our blue carbon other than plantation? Yeah, thank you.
So Yes. So as I mentioned, there are both restoration, that plantation, and conservation. With the restoration, you have to be very mindful that firstly in terms of the methodology, how to restore, because there are studies that shown that after 5 to 10 years, there is additional carbon coming in the ecosystem, which is a study from USA, and they have very extensive restoration over, I think, 20-year period. So it's a long process that requires a lot of money, manpower, and everything. But then there are studies which show that even after 10 years, there is no additional carbon coming in. So it depends. It's not the same like you go in a forest and put a tree. There are definitely some external factors that are contributing, whether even though the restoration is successful, the seagrass would grow, the additional carbon is just not there yet. And that is currently definitely missing in the science. There is no data about it. There are very few studies. We are putting a paper. I'm drafting it right now. So hopefully in a few months it will be out. We visited several restoration sites across Thailand and tried to assess that. Because the studies that have been published are different geomorphological settings, different species were planted, and so on. So definitely there is lack of science there. The science is not there yet. I would just say that. We are trying to catch up. We know that blue carbon is a very hot topic, not only in Thailand, but globally looking, and everyone wants to get their hands on the blue carbon projects. There are some new different global methodologies that involve conservation or biodiversity credits or nature-based solution credits, which you incorporate the carbon, but also other ecosystem services. So you are not relying only on the carbon, but also additional other ecosystem services, which if you go through the conservation project, you would have financial benefit much sooner than the restoration.
Okay, so just the bottom line is restoration is possible. But you need, but you need to have a correct methodology and sign.
Yes.
All right, thank you.
Possible. It is possible, but I would say the science is just tagging behind what the current world wants to do for the restoration for the seagrass.
Hi, Ilka.
Hi.
Right. You hear me, right?
Yes.
Okay. Previously, okay, let me mention that you guys are talking like scientists talking, right? Sorry that I have to let you know that I'm economist. I'm not scientist. Just one simple question for Thailand case here. Suppose that we have a really limited amount of budget in order to have two big projects. One is mangrove restoration. The other one is seagrass restoration. Which one? Which one? Come on. Frankly speaking, come on. In terms of economic perspective, please. Thank you.
Well, in terms of the economy, because I've talked with a lot of blue carbon project economy people, and they know that they want the benefits, the financial benefits. The mangrove projects, because they are currently feasible on much larger scale compared to the seagrasses. So just in terms of that, I would maybe suggest mangrove. But again, I'm a seagrass person, so I'm a little biased for that. But just because the mangrove projects have been proven to be financially feasible so far in many of the countries globally looking, just because of that. But I'm looking forward for the seagrass projects as well, because it is definitely, they really have a potential. They can, we did a simple math in terms of the economy, both mangrove and seagrass. Seagrasses, at some point, mangrove, from the mangrove projects, the financial benefit just stagnates because they accumulate after, I think after 20, 30 years, the carbon in the mangroves just stagnate. It does not increase a lot. But compared to the seagrasses, after 10 years, the carbon in the seagrasses exponentially grows. So in terms of that, it is like, if you are looking really long-term, what we should be looking, the seagrasses have much higher potential, but they are more expensive to restore. And there are other kind of, I would say, negative things. So both of them have positive and negative. So I wouldn't have, if I don't have to pick, but just in terms of the finance, let's just say that.
All right, thank you. No hard feeling, huh?
Okay, thank you. But actually, I would say you need to add seaweed project as well there to think about this. Okay, thank you very much, Monica, to give us a very wonderful talk about this kind of thing, and let me think about the future of blue carbon in Thailand. And we really hope to see not only your paper that come out by the end of the year, but we're also looking for the potential projects about seagrass that can, be a big projects in terms of the blue carbon project in the world. One day we will see the map. Someday, somehow, we will see the map that have the blue circle in the Libong Island that
Yes.
we involved with the projects. Okay,
We are working on that.
that's cool. That's cool. Thank you very much, Dr. Monica from TSU. So the next one, we will move to Kun Panu Bunsongนะครับ from the Thai Union Group, and his talk is about a little bit in terms of the economics. I'm not really sure, but okay, we will see what they are. นั่นตรงนี้แบบเด็กตรงนั้นต่อที่จะมีเอานะใช่ครับนั่นตรงนั้นได้มันมีไมโครโฟนไหมอาจารย์อ๋อได้ครับอาจารย์ตรงนั้นดีกว่าครับผมว่า
power present ตรงนี้ไหม
Okay,ครับ. So this talk is about the overall sustainability strategy of Thai unions, as well as a deep dive into agriculture sustainability and its low carbon practice and technology. A very long topic, but very interesting. So now we set up the presentation first. ใช่ไหมครับ
ได้ไหมนะยังไงไม่มา
Hello. Okay. We have some technical issues. Seems like the cable just got disconnected. Let's see.
ใช่คอมพิวเตอร์มันดับไป
Yeah. Okay.
Okay, please wait for a while. So we now try to set it up. So we still have to talk. One from the Mr. Panu about sustainability strategy, and another talk from our staff. Dr. Noratat will talk about the aquatic animal nutrition. So we still have to talk.
So
Okay.
I think while the team is sorting out the technical difficulties, let me just introduce myself again. My name is Panu Bunsong. I am a aquaculture sustainability for Thai Union Group, right? So my main role is to basically make our aquaculture supply chain more sustainable. So I am involved in lots of projects, ranging from projects focusing on carbon reductions to advocating or supporting the better use or responsible use of antibiotics in shrimp aquaculture. So I'm very, very honored to be here today to be sharing our work at Thai Union. And obviously, having been listening to the scientist presentations has been very, very eye-opening for me as well. So really glad with that. And so the agenda for me today is that, so I'm going to introduce you with the overall sustainability agenda, which we call Sea Change. And then I will provide you a deep dive into our greenhouse gases profile of the company, and then a bit more deep dive into the projects or the research that we are working on at the moment. Okay, I think we're almost there. ได้ยังครับ โอเค so
And Mr. Panu Bunsong has been very kind to our faculty because he attend both program in the morning and having lunch with us and now talk like a scientist. This morning his part is like a sustainability manager. He present about lots of things in the company and stuff. And in the afternoon, I hope that he will be can be more focused on the research element.
Yeah. Well, on that point, I wish I could be a bit more, inclusive in terms of research point of view, but it's just actually starting the research. So what I'm going to do is just to share and to share the background and also the rationale why we're actually pursuing that research and the potential outcome, so to speak. How would that research solve some of the problems that we have in shrimp aquaculture around climate change issues?
Yes, perfect. Thank you. Think we are almost ready.
Yeah, almost. No, the laptop just ran out of battery, so so it needs a bit of time to.
The charger is there but unplugged. I don't know why. Somebody must kick off the charger. Yes. Okay. Perfect.
Perfect. Okay, so let me introduce a little bit about our company, Thai Union. Some of you may have heard it, some of you may have not. But Thai Union is actually one of the world's largest seafood producer. We are based here. We are headquartered here in Thailand, so we are full-on Thai national company. So we have different offices around the world, both trading offices and also factories. So we have the locations are in Asia, in Europe. We have factories in Africa, in countries like the Seychelles and Ghana. And we also have both corporate and also factory offices in North America, like the US and also Canada as well. So our main business is tuna, canned tuna for brands in Thailand, for example, Select Tuna. And we also have business around frozen shrimp as well, which are mostly for export market. And also we are now exploring into new opportunities, for example, supplement business, C-Vita, and also plant-based protein as well, that we are trying to tap ourselves into, also to learn and also to see if there's any opportunities that we can take from there. So in terms of our sustainability strategy, it is called Sea Change, and we have been doing this for since 2016, so for almost 8 years now. Throughout the past 8 years, We are very proud to be recognized by lots of organizations. Notably, we are considered the world's highest performing sustainable companies by S&P Global. S&P Global is the leading ranking organization in the world. So we are very, very proud of that, to be recognized by S&P. And we also try to incorporate new technologies in our supply chain to also advance the traceability and also support us our sustainability endeavor as well. And for the past 8 years, we are part of an organization called CBOS. CBOS is the organization that is initiated by the 9 largest seafood companies in the world. And we work with group of scientists from different countries around the world, for example, Sweden, Australia, to really better the seafood sustainability and also the supply chain of the seafood industry as well. So fast forward to today, right? Back in July last year, we have launched and updated the new iteration of Sea Change Sustainability Strategy, which we call Sea Change 2030, which are which are the goals that we want to achieve in 2030s. So Sea Change 2030 is we plan to help reshape the seafood industry with our sustainability goal for both people and planet. And we are dedicating about 7.2 billion baht, or around 200 USD million, right? So this is the number that we got from the profit that we made back in 2022. So this is a significant amount to be dedicated to sustainability related works within our company. Excuse me. So there are 11 commitments under this new Sea Change strategy, which address 10 UN SDGs, right? So looking back at 2016, there were 11 issues back in the time. There were some of the specific needs that need to be fixed at the times, right? But now, fast forward to today, and we are launching the 2030 goals. So we're actually expanding from the goals that we have set back in 2016. And now we are touching, 10 UN SDG goals. For example, goals like climate action, life below water, decent work, etc. So basically, we're trying to touch everything up on the supply chain that we operate with. So Sea Change 2030 will focus on driving impact for people and for our planet. For our people, we want to protect the safety, the dignity, and well-being of our communities, workers, and consumers. So everything that our business touches upon, right? And then the driving impact for the planet, we really want to see our environment to recover from the climate crisis. And within this two driving impacts for people and for planet, we have what we call 5 key future outcomes, which includes things like human rights, health and nutrition, health and wellness, climate action, biodiversity, and circularity. And again, as I mentioned, there are even our commitments across multiple future outcomes for our people and planet. And I am not going to be able to go through each of them. So if you wish to know more about our strategy, then please feel free to visit our website, seachange.org. and see and also learn more about some of the initiatives and the projects that we have done along the way. So for out of 11 commitments that we have, 3 of them are our marquee commitments, which are the most significant goals that we focus on. But this is not to say that we will not focus on other goals or commitments that we have. These are just what we see as the most pressing commitments that we really need to work on. So the first marquee commitment is called Path to Net Zero. So we aim to have a 42% reduction of Scope 1, Scope 2, and Scope 3 by 2030, and net zero by 2050. And I am proud to say that we are one of the first major seafood company to be for the reductions goal to be validated and approved by an organization called SBTi, or Science-Based Target Initiative. And then the next marquee goal is the responsible aquaculture. So our goal is to have every single shrimp coming into our factory to be produced sustainably. So by sustainably, we mean it has to be either certified or the shrimp has to be in an improvement project, or we call it aquaculture improvement project. And then the next marquee commitment is the responsible wild-caught seafood. So similar to responsible aquaculture, so every wild-caught species coming into our factory, it has to be sourced sustainably, through either certifications or in an improvement project as well. And beyond that, we also require the fishing vessels to be best practiced in both social and environmental aspects as well. So for social aspect would be things like human labor issue for workers on board fishing vessels, and also for environmental aspect. For example, things like reduce or there's no bycatch, for example, on for the vessel supplying wild-caught species to us. Next, I'd like to also introduce you a bit about the greenhouse gas profile of our company. So you see the scale that we need to tackle, right? And this is just one small company, and there are many more companies out there that also need to do the same with us. And I think education, research communities are a very important enabler to really help every sectors to reach the goal of carbon neutrality or net zero, so to speak. So as you can see here, this is our greenhouse gas emissions profile. For Scope 1, Scope 2, we've measured since I think 2019, and since then we have put on some of the measures to reduce the greenhouse gas emissions for Scope 1, Scope 2. For example, installing solar panel across our factories around the world, and also trying to transition from coal to a more sustainable source of energy for the factories as well. And if you look at Scope 3, which is the emissions in our supply chain, right? We started measuring it since 2021, and you can see the ratio here. Scope 1, Scope 2 combined, it's just about 10% of Scope 3. So there are a lot of work for us to do on Scope 3 as well. There are lots of engagement that we need to make, both with the important actors, the suppliers in the supply chain, and also, again, the academic institutions, research communities to find the solutions to enable the reductions in that we have. So here, this slide, I'm trying to drill down into the scope 3, right? And please, this is a bit technical, but please just focus on the category number 1, purchase goods and services, which are the things that we buy into our company. For example, shrimp that we buy from shrimp farmers, or tuna that we buy from tuna trading company, etc. Right? So as you can see, within scope 3, 33% comes from tuna. Well, this is, I think, this is in accordance with the size of the business that we have, because our main business is tuna. So inevitably, we're going to see tuna being the largest in the scope 3. But you see the second from tuna is shrimp, which is around 24%. So I'm going to talk a bit more about shrimp because it is directly under my responsibility, right? But even though you're saying it follows tuna quite a lot, right? 33 to 24. But still, if you look at the whole number, 24% out of scope 3 is around 700 to 800,000 tons, which is more than scope 1, scope 2 combined times 2. So you see the scale, right? That really I really have to tackle and to reduce to bring the company become, say, carbon neutrality or net zero by 2050. There's lots of work for us to do on this one. So I like to also introduce talk a bit around the area of interest around research, to reduce greenhouse gas emissions in the shrimp supply chain. So currently, this is very unfortunate because we are just starting this project, right? So we are currently engaged in a project on direct emission of shrimp aquaculture. So we are working at University of Stirling, which is based in, I think, Scotland, if I'm not mistaken. And also we are working with some of the NGOs called IDH, which is a NGO funded by Swiss and the Dutch government, and also Think Aqua as well. So this project is about trying to address the direct emissions that is from the pond. So basically, it's pond off gassing, right? So if you go into a shrimp farm, you would see. Excuse me. It's a bit cold. Excuse me, sorry. My nose wasn't really clear just now. But for this project, so we are trying to address the gap that is still lacking in the shrimp research around direct emissions, right? So we are basically going to collect gases of the pond using the syringe and some of the tubes that are provided by the University of Stirling. So those equipments are actually waiting for me at the customs of Thailand. So I think the DHL company just called me yesterday to go pick them up. Right, so I think sometimes in the future, once this research is published, then I can share with you more about the outcomes, right? Because we are working with different farm settings and pond settings, right? Is it earth ponds? Is it liners ponds? Or for ponds that has central drainage system and release the feces of shrimp every day, daily, twice a day, etc. So it's going to be, I think it's going to reveal a very, very interesting outcome as well, and also most importantly address the gap in the life cycle analysis or carbon footprinting in shrimp aquaculture. And obviously that is one of the projects that we are currently working on. And also we are interested in expanding the research frontiers, expanding our knowledge to be incorporated in, as one of the interventions, I would say, to reduce greenhouse gas emission in shrimp aquaculture, right? So anything that would promote farming with more efficiency, I think we are interested. For example, we are looking at things like biomass measuring technology. We understand that now shrimp farmers, they sort of estimate the survival rate of the shrimp in the pond, right? And sometimes it's accurate, sometimes it's not, right? Let's be honest about it. And I mean, if let's say if it's not accurate and farmers overfeed the shrimp, and that causes unnecessary waste and therefore higher carbon footprint. So if you have technology that can measure exactly how much shrimp in the pond, and the farmers can easily optimize the feeding strategy, and it become much more precise, and it's going to be more efficient and ultimately lower the carbon footprint as well. So we actually go in a discussion with this company and trying to import the solution here in Thailand as well. But we're trying to see the opportunities how it is going to work here. Also, we're looking at things like upgraded feeder. Right? So as of now, I see that auto feeder, they release pellets in a very narrow area, right? So I am not sure if having an upgraded auto feeder that releases feed pellets in a much wider area would really help in terms of efficiency. So that need to be studied as well. We're looking at things like nano bubbles technology, right? So we know that smaller bubbles can stay afloat in the water longer and can inject more oxygen into the water more efficiently compared to traditional aerations. So therefore, the farmers don't really need to use as much energy compared to traditional aerations to inject an equivalent amount of energy oxygen level. So I think that's something that we like to see as well in aquaculture. And I've heard we've discussed about the alternative feed ingredient as well. So we are also looking at this very topic as well, because things like insect meal or fermentation based ingredients could really help with the reduction of the carbon footprint of greenhouse gas in shrimp aquaculture, right? So we would love to have partners, because we're partners, and so explore the opportunities also to conduct, let's say, even pilot study to gauge that the technologies that I mentioned are viable in Thailand, and most importantly, it has to be scalable as well, because for it to make difference, it has to be scalable. That's about it from me. So would happy to take any questions.
Thank you very much, Kap. So, any question from the floor? Or even people from the online platform, you can ask the question.
Oh, you get a free snack.
If someone asks a question, you might get some tuna free from Mr. Pando. Yeah, for the full year. 2 minutes.
I mean, if you don't have the questions for now, that's fine.
Can I have please some comments?
Oh,
Yes, please.
okay.
Yes, it's very interesting presentations. It's a very big companies, very big, and it's responsible, and will pay more attention on the greenhouse gas. I see they have a project on the shrimp CO2 release. I have some comments
Swimming?
on this. Yeah. We have also have a research on the carbon footprint of the shrimp cultures. And most of the carbon emissions also from the feed and the electricities. I think it's you should pay more attention on this materials, and for how to reduce the carbon footprint of the shrimp, the IMTA I think is a good technologies. MAT means integrate multi-trophic aquaculture. It's a good base. It's just suggestions. Yeah, that's all.
Thank you, Mr. Chang, but I was struggling to hear you a bit. Can you speak up a bit so I can answer your questions?
It's not questions, it's just the comments.
Oh, a comment. Okay, thank you,
Yes.
Dr. Chang.
It's a good idea. Let's we grow more seaweeds and trim together. Okay. Okay, that's it. That's it. Okay. Any questions from the online platform or even in the rooms? Any questions? คุณอัธวุฒิ
มีไหมครับ Okay. Yeah, while waiting for, yes, please, go ahead.
มีคนเปิดไมค์ครับคุณอัธวุฒิเปิดไมค์เปล่าครับ โอเค อาจารย์เชิญครับ
Okay, while I'm waiting for other to have question, I have. You're talking about the outgassing from the shrimp farm. We did not hear this one this morning. So why do you think is a big knowledge gap? What trigger you about how we have to do this? Because this morning we estimate the carbon footprint in the shrimp farm only. The majority is only two, right? It's the energy required for the aeration and energy required to make feed. But you are now talking about outgassing. Does it mean this one is the big portion that has been missing? Thank you.
Thank you for the questions, and this is really good questions. The short answer is we don't know that whether on-off gassing is going to be one of the hotspots in shrimp aquaculture. But what I can say is that when we conduct life cycle analysis, right? From stock hatchery, grow out farms to retailers, that particular area on off gassing during the farming stage is lacking. There's no information, even for research from world renowned universities, in the Netherlands or in the US. So we are trying to address the gap. And if it's going to be, let's say, one of the hotspots, or if it's going to be accounting more than we expected, then again, knowing that is really useful because then we can come up with some of the interventions to mitigate, the emissions associated with on-off gassing of the shrimp aquaculture. So I can share with you more once we have results. So now that research project is just at the beginning stage.
Thank you very much, Kap. So I think we need to move to the next speaker, the last one. Okay, thank you very much, Kap. Okay, Kap, the last one we going to have Dr. Noratat, the Department of Agriculture or Faculty of Fishery Kasetsart University, and the topic today is about the animal nutrition, I think. So please give the Dr. Noratat a hand, Kap.
Okay, where I can find my slide. Stop sharing this one. Let's stop sharing. คุณครับ thank you very much ครับผม good afternoon ladies and gentlemen welcome back to the last session I think the last presenter my name is Noratat Chum you can call me Jump JUMP from the Fish Feed Technology Development Center under the Department of Aquaculture Faculty of Fishery เกษตรศาสตร์ University okay Today, my content of the presentation share. Check. Okay. Hope you guys can see my presentation, right? Yeah. So here is the content of my presentation today. We were talking about the global demand, the food fish and world aquaculture production, and about the CNP composition, and etc. And that all the content that I'm going to discuss with you today. As you can see that the global demand for the animal proteins will be increased about 52% from 2019 to 2050. FAO has expect about the global population increase about up to 10 billion people by the end of 2050. And of course, people need food, and the expensive nutrients is the protein. So the animal proteins will be need to feed the world, okay, for about 10 billion people. And for food, for animal proteins, there are two types of animal protein. The first one is livestock, and the second one is food fish. For aquaculture, we are going to talk about the food fish. Fish is considered as the very high nutritive value of food, or we call healthy protein source containing lots of indispensable nutrients. For example, essential fatty acid like omega-3, DHA, EPA. Okay, and also there are a lot of, the variety of the fish species can be consumed. A lot of variety of the food fish, like from invertebrate and vertebrate, you can see. And also for the food fish has better in the production efficiencies and better in the environment. Regarding to the literature, they mentioned about the fish has the higher in the protein and energy utilization, better food conversion ratio compared to livestock, okay, and higher in the edible meat, lower in the carbon footprints, and lower in the water footprint. All are three aspects that make the food fish has the potential more than the livestock. Okay. So let's come back for the source. The food fish come from two activities. The first one we call captive fisheries, and the second one we call the culture fisheries or aquaculture. By the end of 2030, the production of aquaculture has been expected to be equal or more than the production from the capture fishery. This one is the data from FAO. Okay, and if you can see the current information about the aquaculture, world aquaculture trends, you can see compare between the production amongst the aquaculture and capture fishery. You can see that the production of the food fish from aquaculture has lower than the production from the capture-based fisheries. But the number is not so much different. You can see the capture produce about 90 billion tons, and for aquaculture produce about 87.5 million tons. Okay, and if you can see that the trend of the global aquaculture go to fed aquaculture. There are two types of aquaculture. The first one we call non-fed aquaculture, and the second one is called fed aquaculture. For non-fed aquaculture, we produce a non-fed aquatic animal species, for example, bivalve farming or mussel farming. And for fed aquaculture, we produce the fed animal species, let's say like shrimp, or crustaceans, and finfish. Okay, you can see from the left-hand side of the slide, you can see, I don't know whether you can see it or not, the trend of aquaculture go for the fed aquaculture. Okay, and the last one I calculate, estimate the production of the aquaculture by using the regression analysis, and I found that in 2030 the aquaculture production in 2030 will be about 112 million tons. But regarding to the FAO information, they mentioned about 200 million tons will be up, how can I say, the production into 2030. No? Okay. And if I use this number like 112.4 million tons, I will use this number to calculate or to forecast the amount of the feed that we need to use. I will use the 1.4, the FCI is 1.4 multiplied by 112.4 million metric tons, and I found that 157.4 million metric tons of feed that we need to produce in 2030 for fed aquatic animal species up to the 112.4 million metric ton of the production. Okay. So let's go back to Thailand aquaculture. There are two categories of aquaculture in Thailand. The first one we call inland aquaculture. For inland aquaculture, we produce the freshwater species. And for the second category is the coastal aquaculture. For coastal aquaculture, we use this aquaculture to produce the brackish water species and marine species. Okay, and in Thailand, we have three main products of the aquatic animals. The first one is shrimp, second one is finfish, and the third one is the mollusk. Okay, for the shrimp, is about 40% of the production of the aquatic animal species in Thailand, and for finfish, about 38%, and for mollusk, it's about 22%. For finfish, in this SKU, you can see that consists of tilapia, Charis catfish, and carp. And if you can see the number on the right hand side at the bottom of the slide, you can see I use the number of the tilapia production. Let's say like about 200,000 tons of tilapia need to feed about nearly 300,000 tons of the feed. This one we can use the number of the animal production and to forecast the feed that you may need to use that to produce the animal. Okay, if you talk about the feed, aquatic animal feed, they have to have the three characters. Feed must be edible, must have the benefit to the animal, and also must not be negative or harmful to the animal. There are many criteria to use to classify the type of the feed. For example, in this slide, I use the water content in the feed to classify the feed into three types. The first one we call dry feed. Second one we call semi-moist, and the last one we call moist feed. Okay, for the dry feed, that means any feed that contain the moisture content less than 12%. Okay, for semi-moist feed, That means all of the feed has the moisture content range between 12 to 35%. If the water content has higher than 35%, but no more than 75%, we call the moist feed. Or the moist feed that we found that in the local farm made feed, okay, and also in the blue stock feed. Okay. In this slide, you can see I don't have the pointer here, but you can see that here is the feed. There are many types of the feed. Within of the presentation, you can see this one is the feed for a balloon. And on the right hand side, there are many types of the feed. You can see on the bottom we call boiling feed using for the fishing pot, and you can see a lot of the feed and also some of the marine fish feed. Okay, and if we are using the how can I say the machine to be a criteria to divide the type of the feed, we can also divide into two types of the feed in general. The first one we call the floated pellet, and the second one we call sinking fed pellet. Okay, the floating pellet we are normally use the extruder to produce this kind of feed, for example. So in this slide, that just would like to inform you that there are many types of the feed, depends on the criteria that you use. Okay. And if you guys already know the feed is recognized to be contribute to the significantly emission of the carbon, nitrogen, and the phosphorus through feeding. Okay, and the composition of the carbon, nitrogen, and phosphorus depending on the nutrient content of the feed. For example, the protein content, and also depends on the type of the feed. The feed that you use is the sinking feed or floated feed, and depend on the stage of the animal. In the feed, normally the feed is there are two sources of the pollutant. The first one we call the first one is the feed, or we call unfed feed. That feed that the animal did not consume. Okay, and the second source we call the fecal waste that is come from the feces. Okay, and if you talk about the waste, there are actually two types of the waste. The first one we call suspended solid waste, and the second one we call the dissolved solid waste. This means in the total solid waste consists of two types of the waste. The first one is suspended solid, and the second one is dissolved solid. In these circumstances, some of the aquaculturists, they introduce one of the aquaculture, how can I say, tools to sound sustainable for the aquaculture that we call integrated multi-trophic aquaculture, or IMTA. They said that for the suspended solid waste, this waste will be used to be nutrients for the future feeder animal, like a mussel or mollusk. Okay, and for this soft solid waste, this waste will be nutrients for algae and also for seaweeds. Okay, so they combine together with the fish farm and seaweed farm and also mussel farming together. We call IMTA. Okay, for feed, you can see that feed consumed by fish, we need to improve the retention, I mean the carbon, nitrogen, and phosphorus retention in the body of the fish. And we need to minimize the output, I mean the fecal waste output, okay, so for the how can I say, the waste from the feeding, okay. So this one is the concept for the nutrition. For nutrition, we need we study about the mechanism of the nutrient transformation from the feed to the body. We need to understand the mechanism of that one. So we also propose to environment sustainability sounds that we need to maximize the production by minimize the waste production. That means we need to improve there are some way that we used to improve this one, that is we used to minimize the waste. The first one, we need to improve the feed utilization efficiency. The feed utilization efficiency means we need to improve the food conversion ratio to reduce the FCR, to improve the feed utilization efficiency. For example, to improve the protein utilization, to improve the phosphorus retention, for example. That we can use this concept to maximize the production, minimize the resource inputs, and also optimize the waste output. Okay. And for the nutrition aspect, we need to have the database, because now I and my team study about the nutrient retention since 2014 about the how can we improve the nutrient utilization by using the aquatic animal nutrition and feed technology. We need to estimate the emission of the greenhouse gases. So we need to find out the database. Like in the feed industry, in the feed industry, we have the formulator to formulate the feed. The formulator need the database also to formulate the feed. Or the database we call the nutrient matrix value, that we always need about 70 nutrient metric parameters to calculate and formulate the feed. And also we also need the more database about the carbon and nitrogen and also phosphorus content in the feedstuff and also in the feed also. For carbon, nitrogen, and phosphorus content of the samples involving in the value and supply chain of the feed industry may need to be taken into account. For example, the sample might be the animals, aquatic animals, and the sample might be the feeds and also the feedstuffs. There are many feedstuffs that we are using in the feed industry. There are two types of the feedstuff. No? The first one we call macro ingredients. For the macro ingredient, that means any kind of feedstuff that use more than 1% or more than 10 kilogram per ton feed, we call the macro ingredient. If the feedstuff use less than 10 kilogram per ton feed or less than 1%, we call the micro ingredients. So we need all of the database of that one. And also we need to get the gross energy value of all of the sample. We can use from the calculation, we can use from the analysis. Okay. On the bottom of this PowerPoint presentations, I show you some of the machines that we can use this to analyze the carbon, nitrogen, sulfur, and also phosphorus in the sample. On the left-hand side, we call the carbon, nitrogen, sulfur analysis. I always call this one is a fast protein bomb. Normally, the protein use the the conventional technique to analyze. We have to bring the sample to digest. After digest, we have to distill the sample and have to use the titration. That takes long time. But this technique, we use the combustion to analyze the carbon, nitrogen, and sulfur. On the middle picture, we call the bomb calorimeter. We use this one to calculate to analyze the energy. We can use this machine to analyze the energy. And on the right hand side, we call NIR. In the feed industry, they have the NIR machine to analyze the chemical composition of the sample. Okay, for bomb calorimeter, we can use this one to collect the information about the energy, because sometimes we have the energy footprints, and for on the left-hand side is just the combustion machine. We can use this one to analyze, to collect, to get the information about the carbon, nitrogen, and sulfur content in the sample. For carbon footprint, nitrogen footprint, and phosphorus, sulfur footprint, for example. And also we need to get the information from the animal. Aquatic animal, we have the variety of the species, no? So we need to have the database as well. In this slide, I have some information about the carbon, nitrogen, and phosphorus content in the typical aquatic animal. They said that the carbon content is about 50% of the dry matter in the animal, and for nitrogen is about 10%, and for the phosphorus is about 1.7% or about 17 gram per kilogram of the animal. Okay, so this one is the typical value for the typical aquatic animal. However, we need more because we have many types of the species like crustacean, finfish, and also sometimes we have the echinoderm aquaculture. So we do need to have more information about the carbon, nitrogen, and phosphorus composition of the animal. And of course, we also need more information about the carbon, nitrogen, and phosphorus profile in the feed. In this slide, I show you two types of the feed. The first one on the left-hand side is the sinking pellet. On the right-hand side is the floating pellet, not sinking pellet, it's floating pellet on the right-hand side. For the sinking pellet, I use the shrimp is the representative. For the carbon content in the shrimp pelleted feed range from 370 to about 400 gram of carbon per kilogram of the feed. Okay, and for nitrogen is range from 53 to 71 gram of nitrogen per kilogram feed, and phosphorus is about 1 to 1.56%, or about 10.9 to 15.6 gram of phosphorus per kilogram of the feed. And on the right hand side, I will not read it for you, but you can see the number. I always use this number to calculate the carbon and nitrogen and also phosphorus from the production that I will show you on the next slide. Okay. So we can, if you can see from this slide, From this slide, you can see this one. I show you the application of this carbon, nitrogen, and phosphorus database. We use this one to estimate the carbon, nitrogen, and phosphorus output. Okay, if I use the FCR of the shrimp, how much of the FCR of the shrimp culture? Some of the farmers always tell me that they have the very low FCR is about 1.2, 1.4. However, in reality, when I go and when I visit the farm and I collect all of the information, I got that very high FCR is about 1.6 until 1.8. Okay, I use the FCR is 1.6 to estimate the carbon consumed by this shrimp. I use 1.6 multiplied by 500 gram of the carbon, and then I got that the shrimp carbon composition was about 500 gram per kilogram. That means the carbon from the shrimp. Okay, and if I use the mass balance equation, like input equal to retain plus the output, and to calculate the carbon output, and I found that if I use this number 800 and 500 as I calculate before, and then I will get the number of the carbon output that is about 300 gram of the carbon per kilogram of the feed that we are using. So the question is, if this farm used about 10 tons of the feed per crop, how much of the carbon output of this farm? So you can calculate by using the 300 multiplied by 1,000 multiplied by 10. Then we got that this farm used about 3 tons of the carbon per crop. That's the easy equation to estimate or to calculate. Again, for the floated feed or for the fish feed, we can use the same concept to estimate or to calculate. Okay, like we need to have the FCR of the feed. The FCR equal to feed intake divided by the weight. Again, the weight of the fish. If I need to calculate the nitrogen consumed by the fish, I just only multiply the FCR by the nitrogen in the feed. The nitrogen in the feed is about 60. So I multiply them, and then I got the nitrogen composition about 10 gram per kilogram. And the same way as the previous slide, use the mass balance to calculate the nitrogen output from this crop. Okay, I just put to be your, how can I say, information. Okay, and also there are many factors affecting to the waste output. We need to understand, to know the factors. The factors can be biotic factors like inside the animal, and also biotic factors like environment. Okay, for the biotic factor, for example, like the feed of nutrients like ingestion, digestion, absorption, metabolism, and excretion, all are the factors influencing the waste output for the aquatic animal production. For the digestion, I show you one of the case study for the digestion. There are 4 types of digestion in the animal. The first one we call mechanical digestion, using the mechanical force to reduce the size of the feed. For example, mandible, maxilla of the animal. The second one we call the chemical digestion, using the chemical substances to digest the feed. For example, like hydrochloric in the stomach cells. The third one we call enzymatic digestion, using the enzyme from the digestive tract to digest the feed. And the last one we call the microbial digestion, using the microbiota or probiotic or microbiota in the GI tract to digest the feed. And this one is the case study from my center. We are using the exogenous enzyme to improve the nutrient utilization efficiency and also to improve the digestibility. This study is about 1 month study. Using the exogenous enzyme coated on the feed to feed to the shrimp. There are 5 treatments that use the different ratio of the animal protein and the plant proteins. At the end of the result, we found that the improvement of the ADC, that means the apparent digestibility coefficients. If I use the enzyme that can improve the digestibility of the feed. And for the table 3, I bring the animal to analyze the protein and the nutrient composition in the body. And we found that the nutrient retention increased after using the enzyme. And for the table 4, I used the information from the table 3 to estimate or to calculate the nitrogen mass balance, like how much the nitrogen consumed, how much the nitrogen retained, and how much nitrogen excrete. And we found that the nitrogen retained when we use the exogenous enzyme, the nitrogen retained is increase, is improved. Okay, similar to the excretion. Okay, that if we using the T1, T2, and T3, that I use the enzyme, the exogenous enzyme. You can see that the lower in the digestibility, that means the decrease in the nitrogen retention and increasing in the waste. On the other hand, if we improve the digestibility of the nutrients, that means we improve the nitrogen retention and we reduce the waste excretion. And this case study that I use the insect protein. This project has been supported by NRCT since 2018. We developed an innovative protein source. We investigate the possible protein source in Thailand. At that time, we got the seaweed, we got the microalgae, and also we got the insects. There are three species of the insect that we select. The first one is the cricket. Second one is the superworm, and the last one is the black soldier fly. Okay, this one I select the cricket, the dietary cricket supplement in the feed for you guys to see. Okay, there are three treatment using the different inclusion level of the cricket. Okay, from from 0 to 100% of the replacement of the fish meal. Okay. And then we found that when we using the cricket, the digestibility of the cricket meal compared to the fish meal, the cricket meal has quite good high protein digestibility compared to the fish meal. Is more than 80%. If the protein source has higher 80% of the digestibility, we concern that is the very good quality of the protein. Okay, and when we go and see the nutrient utilization efficiency, we found that the treatment 3, that means the total replacement of the fish meal by using the cricket, improve the nutrient utilization efficiency in terms of the protein retention and in terms of the phosphorus retention. You can see on the bar graph the yellow color. Yellow color does mean on the treatment that we replace, total replace of the fish meal. And also for the table 7, if the feed has the better digestibility, if the feed gives you a better nutrient utilization efficiency, the feed will give you less the waste output. Similar in the table 7. Table 7, we found that the nitrogen total waste from the feeding compared to the control diet. We found that the CM100 and CM50 has the lower in the total waste output of the nitrogen compared to the fish meal diet. Similar to the phosphorus, total phosphorus waste, we found that if we replace the fish meal by using the cricket at 50% and at 100%, the reduction trend of the total phosphorus waste output was achieved. Okay, you can see from also this slide. Okay, and the message from the aquaculture nutrition: if you need to reduce the waste output, you need to improve the feed utilization efficiency. There are many ways to improve the feed utilization efficiency. For example, we need to find out the innovated innovated feed stuff that can use to replace the fish meal. For example, like insect meal, like a bacterial meal. And also we need to improve the digestibility of the feed and the feed stuff by improvement of the technology of the machines, and also by using the feed additives or the feed supplement like the exogenous enzyme. We use this enzyme to improve the digestibility. And also, if you improve the feed utilization efficiency and you improve the digestibility, of course, you also improve the nutrient retention efficiency. That is now the message from, my selected information for you today. At the end of the slide, eating fish, having good health, good mood, and good memory. So let's go and eat fish. Thank you very much.
Thank you very much, Pap. So, yeah, very interesting talk about the feed. I think. Any question from the phone or from the online section at the moment? Okay.
And have very quick question. Thank you very much for the presentation. Yes, this one is like additional for this morning, because this morning they mentioned about the two source of the carbon emission from the aquaculture. One is the electricity used in the farm. The second is the fish or the shrimp feed, right? And they always focus on the reduction or the enhancement of the efficiency of the fish feed, but never talk about the quality or as you mentioned, that how to make sure that the fish use efficiently the food that they take. And that's already like improve the carbon emission, right? Thank you very much. What is the most difficult part? I mean, if you are somebody gonna go for the improving the feed industry, what is the most efficient or the most difficult part to have the less carbon emission? Thank you.
Okay, thank you very much for your question. As my experience, I involved in the feed industry for about 17 years. I was started in the feed industry. My last former position is A&D at the Asian Feed. At the time, also we trying to get more database that we call the sustainable database. One of the database that I would say that the very difficult part, but not so difficult part, is the database. Information and database are the very useful for the feed industry, because the feed industry, they have the program to formulate the feed. We call the BREW, we call the multi equation program, that we use this program to formulate the feed. And we use the nutrient metric, for example, like amino acid profile, fatty acid profile, mineral profile, or etc. of the nutrient. But remaining we don't have is the we call the eutrophying profiles, like carbon, nitrogen, phosphorus in the feed stuff and in the feed, and also in the animal. So I use these opportunities, this chance to encourage you guys to find out, to study more about the database, like a carbon, nitrogen, or phosphorus, we call like a eutrophying, profiles that we can use, because we can use this database to formulate the, how can I say, environmental friendly feed for the future, aquaculture production. That is the first one in for the feed industry aspect. Okay.
So thank you very much, Kap. Any question from Ajahnไหมฮะ?
Not actually the question, but just a kind of unethical comment. And perhaps we have seen you try a lot of things in order to replace like fish meal. You try shellfish, you try insects and everything, right? I think that if we want to solve the problem, down to the grassroots, thing like that, I recommend you try human flesh. Solve all kind of the problem. This should be an ethical comment, but just for fun. And thank you.
Thank you very much ครับ โอเคครับ Thank you very much ดรนรทัศน์นะครับ for the wonderful talk about the feed animal feed นะครับ โอเค Thank you very much ครับ Before we end up the section, we have the quick question that are from the online about any update or projects on mitigating carbon footprint on Thai unions for feed sections. So, คุณ Panu will answer this question.
Hello. Okay. First of all, thank you for the questions, Nakap. Actually, we are working on a project called Low Carbon Shrimp, which I share about this project in the morning session, but I didn't in the afternoon section because we actually focus on research. But within that project, there are a number of interventions, both farm and off-farm interventions, right? So for off-farm interventions, we are focusing in the feed, right? So we are going to incorporate 100% of deforestation and conversion-free soil to be used in the feed and also to be used as part of low carbon shrimp project. So using DCF, deforestation conversion-free soil, will reduce the carbon footprint of feed and also ultimately reduce the carbon footprint of the shrimp product. That's the project that is going on at the moment. But research speaking wise, we are also trying to explore the valid alternative feed ingredients that would be replaced, let's say replaced on things like fish meal, right? So we are working a lot on that as well. So we are talking with lots of startups, research institutions to look for viable and more importantly scalable options to be used in our supply chain. Thank you.
Thank you. Okay. ครับ Thank you very much ครับ คุณพานุนะฮะก็ I think this is we come to the end of the seminar today. So thank you very much for speaker that provide us very informative presentation, very useful and inspiring thing that I think we have some idea from the talk for sure. But anyway, still need to go along the way with a thing to improve. I think it's kind of jigsaw of the thing that's one people talk about one thing, another people talk about another thing. So for better life, we need to incorporate everything, and we really hope that the Faculty of Fishery will be the main institute to try to improve something, right? Okay, I think time is up and I talk too much. I need to pass this back to the MC. So thank you very much for joining this art, and thank you very much.
Thank you very much. Yes, hello again. Thanasphong UMC is here. I have a very, very 3 hours. It's kind of passed very quickly. Thank you very much for the moderator and also for every presenter. So for Professor Chang and Dr. Milika, if you're still with us, and also Dr. Kong, maybe you can turn on your camera and join us at the end of the ending ceremony of today's seminar. With this, I would like to invite the Vice Dean for Research, Associate Professor Dr. Wanchai Watanametikun to present the token of appreciation. First to our speaker, Kun Panu Song.
Okay, thank you very much. Yes.
And second is Dr. Norathat Prachum ครับ And also to our great moderator, Dr. Valantat Bulyapuk ครับ and Associate Professor Dr. นรงค์ฤทธิ์เมืองใหม่ครับ All right, and last but not least, Professor Chang and Dr. Milika. Dr. Milika will be sending our token of appreciation to you. And Professor Chang, we are waiting for you to visit Bangkok and to present you the token of appreciation. Lastly, can we take a group photo together? Can you please move on to the point? Okay, so please wait a moment. We will try to set up the screen. I will also run to join. Okay.
Yes, you can email the photos for us.
Can mail to you for sure. Thank you very much. Thank you very much, everybody.
Thank you.
Thank you very much.
It's nice meeting you. Thank you.
Thank you. Yeah. I'll leave the meeting.
ได้เลยครับว่าท่านอื่นต่างไปคิดมากเลยครับขอบคุณครับ
Thank you very much ครับ Dr. Milika and Chang Lao Sư. Thank you very much.
Thank you. Bye bye.
Bye bye.
Bye bye.
Bye bye. 再见再见。
再见。
ตามมานะ